microcolony. So this is the review for the practical, the first practical, and I'm gonna go ahead and record this for you, and then you guys can take a look at it at your convenience, and then we'll talk about it when we meet up again. Okay? So I've got a few questions to go over with you, but I wanted to tell you a little bit about the setup. So if we look at this particular slide, you'll see that there's a question, and it has to do with colonial morphology, right? So you'll see the question is written right here, and then there'll be some directions, right? So this says for number one, you want to describe the margin, right? So you want to come over and look at the margin, and you can see that that particular margin is serrate. Somebody might call it irregular. And so here you can see these little boxes, right? So these little boxes, you would type in serrate or irregular, okay? And then the second one, it's talking about elevation. As you come over here, look at the elevation, and you can see that that particular one is coming up off the media, right? And so you can say that it is convex, and so you'll see it's convex. It's not umbonate, so if you wrote that down, it would be incorrect. So if you think about that for just a little bit, be sure that when you put these words down, right, that number one, I want you to only use lowercase letters, because the system, the way I've got it set up, is that I'm going to ask it to not be as specific. So if you leave it lowercase, then it's going to be easier. So I'm not going to count off for spelling, but you have to get it close. Like for instance, if you thought that an image depicted a dog and you spelled it D-A-W-G, that would be okay. But if you thought it was a dog and you spelled it H-O-R-S-E, then that's incorrect, right? So I'm gonna stare at that particular word for about 2 seconds and decide if it is close or not. And then if it's close and it looks okay, then you'll get credit for it. If it doesn't look correct at all, you just won't get credit for it. Now, the system will grade most of it for me. I just need to go in and look at the ones that it's got questions about, and it will flag all the incorrect questions for me so that I can go in and quickly look at them. Okay? So if you have questions about the way it's going to be all fill in the blank. There'll be 50 questions. There may be some EC, right? It'll be randomized, so nobody's going to have the exact same test, right? So, and if you have 50 questions, you'll have 52 minutes to take the exam, and then the exam will be over. And you'll get directions from me. Okay, we'll talk about this when we meet in class again. All right. So if you got questions, then you can give me a holler, or we can talk about it when we get back together. All right, let's talk about the review, right? So here, appreciate the slide, right? And so you're looking at these particular structures that are staying purple, right? And so on an exam, I will always tell you what color it is if it's not discernible, right? If I think there's any way that might be a little bit pinkish or reddish or whatever, and there's not an exact color, then I will tell you. Here, there should be no mistake that is purple. So question number one, identify the gram reaction at the pointer. So you could say gram positive, you could say GP, right? But you cannot say G plus, because the system will count that wrong. And I'm telling you right now, that is not a correct way to denote gram positive. If you do that, it is incorrect, and I will count it wrong. You have to spell out gram, G-R-A-M, positive, P-O-S-I-T-I-V-E, or you can put GP, right? But you cannot put G with a plus sign, because if you do that, that's incorrect. We're going to use the correct vernacular, and that is not correct vernacular. Okay? Okay, so that's the answer for the first one. What's the difference between gram positive and gram negative cell walls? Lots of ways to answer this, right? So you could say gram positives have a thick peptidoglycan. That's correct. You could say gram negatives have an LPS layer. That's correct. You could say gram-positive have teichoic acids. That's correct. You could say gram-negatives do not have teichoic acids. That's correct also. You could say gram-negatives have a smaller peptidoglycan than gram-positives, and that's correct. So there's lots of ways to get this particular answer correct. All right. Anybody, if you've got questions, let me know. Okay. Number 3, identify the arrangement at the pointer so it is in chains or it is strepto. If you say cocci in chains, that's correct. If you say streptococcus, that's also correct. Okay? And name the counter stain used in the gram stain. Remember, the counter stain is the second stain used in a staining methodology. So the counter stain in the gram stain is safranin. Now, one thing I want to tell you all is that if you're looking at a slide and you see purple or you see red, you assume it's a gram stain unless I tell you differently. Okay? So that's one thing you should always remember. Purple and red, gram stain unless PRoV says differently. You should also know that if you see green and red, that it's an endospore stain. If you see black and purple, then it is a capsule stain. And if you see hot pink and blue, then it is an acid-fast stain. Right? So if you know those things, then a lot of this exam is just going to be really, really easy. Right? So let me repeat that again. Red or purple, if you see that on the slide, then it's a gram stain. Assume. If I don't tell you anything, it's a gram stain. If I say this particular stain is a simple stain, then that's different, right? But assume if you see any purple or any red, it's a gram stain. If you see green and red, then it's an endospore stain. If you see purple and black together, then that's a capsule stain. And if you see hot pink and blue together, it's an acid-fast stain. Okay? Next questions. So here again, you're looking at purple and red. And so if you follow the rules, purple and red, you're looking at green and red. Green and red. So if you're looking at green and red and are following the rules, then you immediately know that this is an endospore stain, right? So identify the special staining technique. It's an endospore stain. What is the main function of the structure that's green seen at the pointer? And that's survival, right? And identify the primary staining reagent, right? Primary is what comes first, and so that would be malachite green, right? So while those are some 3 questions, I think could be asked about the red structures, they could be asked about the decolorizer, they could be asked about the counter stain, there's lots of things that could be asked about this particular image, right? What is the composition of the outer layer of the green structures? What's the gram specificity of this particular organism? All these things should be known because we've already talked about them, right? And by the way, all endospore forming rods, by definition, are gram positive. Okay, let's go on. So you see purple here, right? So you're assuming it's a gram stain. Identify the staining technique. It's a gram stain. Identify the purpose of the iodine, number 9. Identify the purpose of the iodine and this technique. It's a mordant. You could say it's a fixer. You could say it's a sealer. All those are fair, right? Name the primary morphology seen in the slide. It's asking for morphology. It's not asking for arrangement. So in this case, the answer is coccus or cocci, right? And true or false, the bacterium in this field of view is motile, and the answer to that is false. By definition, all cocci are non-motile. Okay? So here is, of course, the streak plate method. It's sitting on 2% agar, right? That's what's in the media. And so those things can be asked about. Those are all very fair questions, right? Identify the culture method. That's a streak plate. What's the purpose of this method? Be very specific. It's to isolate colonies, right? What is the object at the pointer called? That is a colony. What is a CFU? Number 15, what is a CFU? That's a colony forming unit. And then what tool is used to conduct this method? And that would be an inoculating loop. You could just say loop, that would be fine. But if you said needle, then that's typically not going to be correct, right? The typical tool that's used here is an inoculating loop. Okay, let me know if you have questions. You can call me, email me, we ask in class. Again, you're looking at purple elongated structures, right? So what is the gram specificity in the morphology here? You can say it is a gram-positive bacillus. You can say it's a gram-positive rod. You could say GPR. You could say GPB. Okay, those are the only correct ways to do this, right? Okay, untrue or false, the bacterium in the field of view contains an LPS layer in its cell wall. That is false. It's gram-positive, therefore it only has a peptidoglycan layer. Okay. So here you can see this is a flagellar stain, right? Identify the staining technique, and the staining technique is a flagellar stain. Is this organism motile or non-motile? You would say motile, right? And what's the arrangement of the flagella? What's the flagellar arrangement in the field of view? So you can see that there are multiple flagella, a tuft of flagella, at both polar ends, right? Even though you can't tell it at this other end, if you have a tuft of flagella here, you're going to have a tuft of flagella here, right? So therefore, this is Lophotrichus. Be sure you know the difference between Lophotrichus, Peritrichus, Amphitrichus, Monotrichus, and Atrichus, right? Those are all fair questions to be asked about. Okay, next question. So here again, you're looking at motility media. It is of semi-solid consistency. It has approximately 0.5 to 0.8% agar in it, and that gives it a consistency of jello. We say it's a semi-solid. So number 21, what's the consistency of this media? Well, it's a semi-solid, right? What tool is used to inoculate this medium? And of course, that is an inoculating needle. You could say needle on just needle. You could say straight needle, that would be fine. But I don't want to hear a pokey thing, or I don't want to see somebody say a straight thing. I don't want to, you need to tell me it's a needle somehow or another. Okay? If you say it's a loop, it's not correct, right? A loop is not used for this technique. Identify which tube of the three, A, B, or C, illustrates a non-motile organism and why. Well, that would be A, right? And that's because you see that the site of inoculation has got growth in it, but there is no movement of growth away from that site of inoculation. That's almost a perfect inoculated or motility medium. The other two show that there is growth because of the fuzziness that we see in both of those tubes, and those are both motile. So the answer for A is non-motile. The answer for B and C are motile. There is not an answer. It's a little motile. It's a binary. It's either motile or non-motile. Okay, and the term is motile. M-O-T-I-L-E. It's not mobile. That's incorrect. If you say mobile, that will be incorrect, right? That's a whole different word. It means something very different. Words have meanings, and while I'm a little bit lax with spelling because of my dyslexia, I will not give you credit for things that are incorrect. So M-O-T-I-L-E is correct. M-O-B-I-L-E is not correct. Okay? Let me know if you have questions. So then here's the microscope, and you can see that there's some questions surrounding the microscope. Number 24, identify the microscope part labeled 1. And so it's right here. The answers that are correct are either eyepiece or an ocular, either one of those. Okay. What's the magnification of the part labeled 1? That is 10x or 10 times, right? And so remember that not only is this the first magnifying lens, but it also has the function of bringing the field of view to your eyes. Identify the 26, identify the microscope part label 4. So you can see that 4 is pointing to the nose piece or the revolving nose piece, right? And so then number 27, what is the magnification when using the oil immersion objective lens? And so these are objectives, right? And you're using the oil immersion, which is on the other side. You can't see it, but it's labeled 100x. So it's total magnification because that's what's being asked. What is the magnification when using the oil immersion lens? It's 100 times 10, which is 1,000. Okay. Oh, okay. So I'm making that distinction. So 28 is 1,000, and 27 is just looking at the oil immersion objective by itself, right? So the oil immersion is 100 times, and therefore 100 times 10 for number 28 will be 1,000. I'll be sure to clarify those so that you won't make the mistake that I just did. Okay, let me know if you have questions. Now, I can ask about any of these parts, right? Um, so be sure you know the parts of the microscope and its function. Okay. All right. The last grouping has to do with colonies growing on a plate, right? And so here, this goes back to day one, right? What is the origin of the structure labeled A? So if you come here and look at A, you can see that it's glistening, right? It's kind of tannish in color. It looks a little bit like it's mucoid. So the answer to this would be the origin would be it originates from water, right? So what's the origin of the structure labeled B? You come back here over to B and you see that it's rough, it's fairly large. And so therefore, this is originating from soil, right? A yellow structure, like some of these yellow ones in here, right? They would originate from people or animals, right? These yellow ones, or these little white ones also, right? This large, big structure, you have to be careful because if I ask the question, if I had labeled this colony here, and I would have said something, describe that particular colony, then you have to understand that that colony is all of this, right? All of this is that one colony. So you have to understand the way that colony is formed, right? And so I might ask questions about what is its margins, and you would say, well, those margins are highly irregular, right? What's the size of the colony? It would be large, right? And then, of course, I might ask about where did that particular structure originate from? And because it's rough, it's likely to originate from soil because that is a bacillus, right? And when I say bacillus, I mean that that is the genus, therefore it should be underlined. Now, when you write this bacillus, and I'm not going to ask you, I promise, I will not ask about genus and species on the first practical or the second practical, right? But I'm just making note that if you see that, that's genus, where if you see this, then that is just simply a shape. Okay. So everything that we've talked about in Unit 1 is fair game. There'll be questions about ubiquity. There'll be questions about hand washing. There'll be questions about fluid thioglycolate. There'll be questions about candle jars and anaerobic jars. There'll be questions about ambient conditions. There'll be questions about um, media and the percentage agar that's in a solid, 2% versus a semi-solid, 0.5% versus a broth, 0%. What percent of agar is in thioglycolate, 0.05%. So all those things we talked about. You could look at everything that is in that first practical unit, and there'll be questions about every single thing. I have over 100 questions written. You'll get 50 of them, right? So nobody's exam is going to be the same. Everybody's exam will be different. Okay. So this is the end of the review, but just think, all the stains, all the media, all the tools, safety, all those things we've talked about. Again, take a look at that. Take a look at the lab schedule, and everything on there that's prior to practical 1 is fair game on this practical. It'll all be fill in the blank. There'll be 50 questions. You'll have about 52 minutes to take it, and then we will have lecture afterwards. Okay. When we meet up again, then you are, I'll, we'll have a lab, but at the end of that, you can ask questions to clarify anything you might have had a question about on this review, or anything that has to do with the lab. Okay. So let me know if you have questions. Either text me, email me, or you can ask in class when we meet again, and then we'll clarify those things so you'll be ready for the practical. Take care. Bye bye. PRoV out.